I'm Dr. Madhu Nag, and I serve as the Chief Scientific Officer for Inspiro. Oncology research really suffers from lack of reproducibility, and I think ARCTis really is based on the model of excellence for us at Inspiro, which is robustness, reproducibility, and scalability. So by offering researchers the ability really to have the same results time and time and time again, and not have that lot-to-lot variation from researcher to researcher, day to day, you're really ensuring the robustness of your results. And then when it goes on to the next stage, you can have confidence that what you discovered in that stage was valid. Typically, in early discovery, you want to test multiple cell line models before going into patient-derived models. And being able to do this time and time again, both in a primary screen as well as in a secondary validation screen, becomes very important. Being able to reproduce that data becomes very important. And I think this is where ARCTis is going to find its home, not only among high-throughput screening centers, but within every discovery lab with big pharma and biotech. So the FDA Modernization Act 2.0 really set the stage to introduce people to new approach methodologies, which includes more evolved in vitro screening methods or in vitro tools. And the FDA Modernization Act 3.0 takes it a step further. And here I think there are multiple perspectives. From the perspective of the researcher, ARCTis brings to the table, again, the ability to generate the same data over and over again so that you're able to move from early phase to sort of late stage drug discovery. On the other hand, from the point of view of the regulators, they receive data that is robust and reproducible and that they can use to qualify in vitro methods. And this has been sort of the gap in the adoption of new approach methodologies from the regulators' perspective. And I think that's where, again, the ability to build in reproducibility is what ARCTis's sort of unique selling proposition is. For anything that requires high throughput screening, the ability to build in complexity and not have variation that you would see if you were doing this with multiple cell lines over a period of time is invaluable. So if you can imagine, with the ARCTis portfolio of just cancer cell lines, you get the therapeutic effect of any drug on any cancer cell line. And then you build in the complexity by the addition, like sort of the tiered addition of multiple different cell lines, and you're able to tease out the effect of multiple different therapeutics on different cell populations, which really then gives you a very good idea of what the therapeutic window of any therapeutic is on a multi-cell type model, which is a very good indicator of how that drug will work in vivo.